Hello students, welcome to today's Life Sciences lesson for grade 12. And we have a question and answer lesson, and it's based on DNA replication. So the first question that we have has a diagram, and it says the diagram is representing DNA replication. So let's look at our diagram very carefully. What we can see is a double-stranded structure. And then what we also see on the diagram, if you look at the left, is that that double strand has separated. So you actually have a gap in between. If we look more closely, we can see that a single part is highlighted, and it's got the labels X, Y, and thymine. And the entire thing is circled, and it says molecule W. You've got nitrogenous base as a label. You've got a bracket with the label U right at the end. You've got another nitrogenous base at the bottom that's labeled. And at the top, the first little label we have pointing at one of those little blocks has the letter V in it. Okay? So look at it carefully. And we've got the letter Z as well, right? So look at that very carefully, and let's look at the questions. So the first question is, identify the following. And it says molecules W and U. So if we look at molecule W, right at the top, you can see it's made up of three parts. There's the X in a circle, there's a Y in a pentagon, and then in a rectangle, you've got the word thymine. So what is that collectively? That's a nucleotide, which we know is a monomer of your all nucleic acids, right? If we look at the next one, label U, that's right at the end, and there is a bracket. So that's bracketing off the two strands together. So that label would simply be DNA. Right? That's your double-stranded DNA. The next question, identify the parts of molecule W that are labeled X and Y. So X in the little circle would be phosphate. That's your phosphate group. And Y in the pentagon would be deoxyribose. Just remember, it's not good enough to say sugar, or it's not even good enough to say pentose sugar. You have to specifically tell us which one. So the only answer for Y is deoxyribose, right? The next label we have to identify is bond Z, right? And what can we see? If you look at Z, you can clearly see that bond, or those bonds in that region, are holding the two strands of DNA together. And the only bond that does that is your hydrogen bond, or you could say weak hydrogen bond. Right, the next question, nitrogenous base V. Now, if we look at nitrogenous base V, we can see it's complementary to the nucleotide that's highlighted. And that nucleotide has the base thymine. So you've got to ask yourself, so what's complementary to thymine? That will be V. Complementary to thymine is adenine. Right, so your answer there is adenine. Right, the next question. Where in the cell does the process take place? So where does DNA replication occur? It occurs in the nucleus of the cell, because that's where our main DNA is found, if we can say main DNA. Right, our next question. Name the phase of the cell cycle where DNA replication occurs. So it occurs during the interphase, right? So during the interphase, remember, is before the division. That's the part of the cell cycle. And if we look at our last question, notice that one is for 6 marks, right? So let's look at what that question states. It says, discuss the process of DNA replication. Now, when you have a short note or part of an essay, it's very important that you are able to describe a process in a logical sequence, right? So we can't go back and forth, right? It's got to start at a certain point and it ends up at a logical conclusion, right? So let's look at what we can say about the process of DNA replication. Firstly, you could state that DNA replication is controlled by enzymes. The next point, your double helix unwinds. Next point, the two strands of your DNA separate from each other when the weak hydrogen bonds between them break. So you can say the hydrogen bonds are breaking, or you can say DNA unzips, but you've got to say then the strands separate from each other. Then what do we say? Free nucleotides that are in the nucleoplasm are going to attach themselves to the two strands of DNA that are formed. Each strand of DNA is acting as a template. So you get free nucleotides that become attached to complementary bases on each template. You then will have two new and identical molecules of DNA that are produced, and these DNA molecules will then coil up and form a double helix again. So maybe that answer was worth more than 6 marks. But remember, in a question of this type, you rather discuss the process fully. State more than 6 points if you are able to explain the process in more than 6 points. So right, students, let's look at the next question. It says the table below contains a list of statements about DNA replication. Some of these statements are incorrect. So you need to state which statements are correct and which ones are incorrect. So let's look at our table, right? So the first statement is the DNA molecule unwinds. So that statement is correct, right? The next statement, hydrogen bonds between the base pairs break. That's correct as well. And notice these statements are going in the sequence of the process of DNA replication. The next statement, free RNA nucleotides join to the bases on the exposed DNA strands. That statement is incorrect because if you read it carefully, and please read carefully, it says RNA nucleotides. It should actually say DNA nucleotides. So that's an incorrect statement, right? The next statement, only one strand of DNA acts as a template. That's incorrect, right? Because in DNA replication, both the strands of DNA act as templates to build their complementary strands. The next question, or the next statement, hydrogen bonds form between the complementary bases. That statement is correct. And the last one on the table, 3 hydrogen bonds form between the bases T and A. That's incorrect. Thymine and adenine are bonded together by 2 hydrogen bonds. It's your guanine and your cytosine that have 3 hydrogen bonds, right? Okay, let's move on to our next questions. And let's look at multiple choice. So the first question is, and read carefully. First question, which of the following processes occur during DNA replication? 1, breakage and reformation of sugar phosphate bonds. 2, breakage and reformation of hydrogen bonds. And 3, alignment of free nucleotides with their complementary bases. Right? And our options are A, 1, 2, and 3, B, only 1 and 2, C, only 2 and 3, and D, only 1, right? So let's look at those statements again. So the first one says breakage and reformation of sugar phosphate bonds. That does not happen in DNA replication, right? Remember those parts of the strand, the backbone of the strands remain intact, okay? So that one's out. So if you look at option A, that will be out because 1 does not apply. Option B is going to be out. Option D is out as well. So the only option we're left with is C. And let's see if that's accurate. It says 2 and 3 only. So 2 says you get a breakage and reformation of hydrogen bonds, right? And that's true. They first break, so your original strands separate from each other, and then they reform when your new DNA molecules are formed. And if we look at 3, the nucleotides aligning, coming in line with complementary bases, that's certainly true as well. So your option here is C, right? So let's look at 3.2. Replication is when A, proteins are made, B, RNA is made from a DNA template, C, another copy of DNA is made, and D, DNA is made from an RNA template, right? There's only one correct answer, and that one is C, another copy of DNA is made. Right, our next question. Which is true about the pairing of bases in the DNA molecule? A, purines always pair with pyrimidines. B, a single ring base pairs with another single ring base. The next one, a double ring base pairs with another double ring base. And D, purines pair with purines and pyrimidines pair with pyrimidines. Now students, to answer this question, you need to know what's a purine and what is a pyrimidine, right? So remember the purine, the examples of purines rather are adenine and guanine, right? They are large, they are made up of 2 rings in their structure. Your pyrimidines are the smaller bases. That's your thymine and your adenine, and they are made up of 1 ring, right? So let's see which ones apply. Here we're talking about pairing, right? Complementary. So we know adenine and thymine pair together, and guanine and cytosine pair together. So which is your option? Your option is A. Purines always pair with pyrimidines, right?